Hello learners, welcome to Ahmad Coaching. Today we are going to study about law of independent assortment. In the law of independent, the independent mean that they are not controlled by anything or they are not dependent on anything. While the assortment is the sorting of things into the groups. For example, you have 3 red shirts and you have 3 black shirts. So what you are going to do, you are just going to arrange the shirts according to their number. So it means that you are assorting or you are sorting them into the shirts. Now here the assortment means that if you are studying a dihybrid cross, dihybrid means that you are studying 2 traits. For example, I am studying that a person has a hair color and a blood color. So I am studying dihybrid cross. If I am going to study just the hair color, then I am studying the monohybrid cross. But if I am studying hair color and blood type together, it means I am studying dihybrid cross, or I mean 2 traits. So Mendel proposed this law of independent assortment for the alleles of dihybrid cross. In my previous lesson, I have already told you what alleles are. The alleles, they are the partners of a gene which are present on the homologous chromosome. For example, the alleles for the eye color is present here as well as here. So these are the homologous chromosomes and these are the alleles. So according to this law, when 2 contrasting pairs of traits are followed in the same cross, it means that when you are studying 2 different traits, for example hair color and blood type, in the same cross, their alleles assort independently into the gametes. So their alleles which are present on the different homologous chromosomes, they are going to be separated independently into the gametes. It means that they will not say that, okay, I was present with you on this homologous chromosome, so let's go together. No, this is wrong. This allele has its own will. This allele has its own will. It can move here and this one can move here. So they are not dependent on each other. They are independent and they can go to any gamete they want. Okay, so this is what we call the law of independent assortment. For example, here I have round shape yellow seeds. So this is one trait and this is another trait. So in this plant we are going to study 2 traits, or we are studying the dihybrid cross, in which I have round shape seed which it has yellow color. So for the round shape, capital R, while for the yellow color we have capital Y and capital Y. If I'm going to cross this plant with wrinkle shape green seed, so here we can see that small r representing wrinkle shape and small y representing green seed. So this is P1 generation, and in this P1 generation we have 2 traits we are studying. So what will happen, that this capital R, it will move to one gamete. Similarly the Y will also move to the gamete. Here, because both the alleles are same, so we will have capital R and capital Y. Similarly here we will have small r and small y. So these are the gametes. Now in the random fertilization, this gamete is going to fuse any of the gamete from the second parent. For example, this capital RY will fuse with the small ry like this, and we will have capital R small r capital Y small y genotype. Okay, always remember, although the gametes they are like having capital RY like this, if you see them, but when we do this we are going to arrange these according to the alleles. For example, capital R and small r, capital Y and small y, so that we know that this pair of allele is for one trait and this pair of allele is for another trait. So again, this one is going to fuse with the other gamete and we will have this genotype. This one will fuse with this one and we have this genotype. Again, this one will fuse and we are having this genotype. This is F1 generation. We will have heterozygous combination here. Now, for example, in the next generation, I'm going to cross any two plants having this genetic combination. Now you can clearly see how these alleles are independently assorting from each other. Here we can see that this capital R allele we have here, and this allele is moving with this capital Y. Okay, we saw this in this case also, but this capital R can also move with the small y because it is independent. It is not necessary that capital R will always go with the capital Y. It can also go with the small y. Similarly, the small r, it can also go with the capital Y, and it can also go with the small y. Why? Because they are not dependent on each other, they are independent. Similarly, here we also have the capital R with the capital Y, capital R with small r with capital Y, and small r with small y. So these are the gametes. Now, if they are going to fuse with each other randomly, we can see that this capital R, it can fuse any of the gamete from here. So for example, this first one fuses with this. We will have capital R and capital Y and capital Y, but this gamete can also fuse with the second one. So here we have like this. So we have the capital R, small y and capital Y. This can also fuse with the third one like this, and we will have capital R, capital Y, and from small r and capital Y. So this way they can also fuse with each other. But as you can see, this is like a jungle here, and it is going to be very difficult to understand what is actually happening. So make it easy, we are going to use the Punnett scale. In the Punnett scale, at one side, or you can say that at x-axis, we are going to write down the gamete of one parent, and on the y-axis we are going to write down the gametes of another parent. So here we have capital R, capital Y like this. We have small r, capital Y, this one. We have small y and capital R, this one. And we have small y and small r like this. Similarly, these are also here. So how we are going to do this? This one gamete is going to fuse with this one. So capital Y, capital R here, and capital R from here. Similarly, this can also fuse with the other one. We will have this genetic combination. Similarly, this one can also fuse with this one. So we will have capital Y from here, small y from here, capital R from here, and capital R from here. Similarly, this gamete can also fuse with this one. So we will have capital Y from here, small y from here, capital R from here, and small r from here. Similarly, the second gamete will fuse with all these four gametes like this. Third gamete will fuse with all these gametes, and then fourth one will also fuse with these four gametes, and we will have the genotype. Now, according to the genotype, we can see that whenever there is capital Y, it means it is dominant, and whenever there is homozygous recessive, it means it is recessive. So we can see that capital Y, capital R, and capital R mean that the seed is going to be round, and capital Y mean the seed is going to be yellow. Okay, similarly, here we can see that capital Y mean the seed is going to be yellow, but small r mean that it is going to be wrinkled. So we can see that it is wrinkled. Similarly, here we can see that small y mean the seed is going to be green, while capital R and capital R means that it is going to be round. So we can see that we are having new combinations in the F2 generation. And why we are having new combination? Because the alleles, they are independently assorting from each other. They are not sticking with each other, but they have their own will, so they can go to any gamete according to their own choice. So in this way, we will have 9 yellow round seeds, 3 green round seeds, 3 wrinkled yellow seeds, and only 1 wrinkled green seed. So this is the phenotypic ratio of our law of independent assortment. I hope this lesson is clear to you. If you still have some question, you can ask me in the comment section. I will see you in the next video.